Okay.
Good afternoon, everyone, and welcome to the Native Voices in STEM seminar series, which is co-sponsored by the University of Arizona Sloan Indigenous Graduate Partnership, as well as the NSF NRT Indigenous Food, Energy, and Water Security and Sovereignty Program. We're honored today to be hearing from Dr. Otakuye Conroy-Ben, who is from the Oglala Lakota Nation. She is an assistant professor in the School of Sustainability and Engineering and the Built Environment at Arizona State University. Dr. Conroy-Ben received a Bachelor of Science in Chemistry from the University of Notre Dame, as well as a Master's of Arts from the University of Arizona in Analytical Chemistry. Here at the University of Arizona, she also received her PhD in Environmental Engineering from the University of Arizona. Another important highlight that is pretty awesome is that she's a UA Sloan Scholarship alumni. And she's also the recipient of the American Indian Science and Engineering Society Technical Excellence Award for this year. So please join me in welcoming Dr. Otakuye Conroy-Ben.
And I thank INDIGE-FEWSS and the Sloan organizers for inviting me to speak. I always like an excuse to come back to U of A. I have been here still. And I must say the campus has changed. Coming down Park and Euclid, I don't recognize the surrounding areas very much, but it's very nice to be back. So I understand that this seminar series is for the INDIGE-FEWSS scholars as well as the Sloan scholars. And I see some of my former faculty members here. So I'm going to talk about kind of my journey into the professoriate, and then get into more of a technical talk about my current research interests. So this slide kind of summarizes what I do. I look at emerging contaminants arising from wastewater pollution. They really started here at the University of Arizona in Dr. Bob Arnold's lab, where I was looking at endocrine disruptors. And this was back in the year 2000 when it was really an emerging field. But there are other novel contaminants that are still being studied. So there are drugs, antibiotics that can lead to antibiotic resistance, substances of abuse, as well as chemicals on these Teflon cans. And so my talk is going to kind of focus on this area of emerging contaminants. So my research interests are really broken down into three main areas: environmental endocrine disruption, antibiotic resistance, as well as wastewater-based epidemiology. And this is a nice little diagram which shows the water cycle from an engineer's perspective. We have waste, okay, coming from humans, and that can go into either solid waste, water, or into the land. You can see that gets back into our water sources, and then it gets back out into our water supply, and then eventually back to us. So my interest in research started before I became a graduate student. I was a lab tech at the University of Colorado, where I got a lot of experience in genetic sequencing. And this was sequencing back in the late 90s, which is outdated technology. But what came of this was a deep respect for rigorous scientific methods. Also coming from this is I was able to appear on some publications which really set the way, set the path for my research career. And then came U of A. I spent actually quite a bit of time here. doing my master's and PhD, and in different areas. And then once I graduated, I had many different career options. So this was before I came to the University of Arizona. Once I graduated with my PhD, I had a number of different offers. I ended up at LA County Sanitation Districts as a wastewater engineer. Dr. Bob Arnold introduced me to his former colleagues there. When I was there, I looked at odor mitigation and sewer, so sewer odor, which is very interesting. But I missed the freedom of academic research. So I came back to the University of Arizona, and I did a postdoc in biochemistry and environmental science. So I was in SWES and in biochem. While I was here, I was adjunct at Pima Community College. And then I went on to the University of Utah for about 4 or 5 years. And then I applied to Arizona State University when they had an open faculty position, and that's where I am now. Okay, so I'll get into more of my technical talk. First, talking about endocrine disruption. Who has not heard of endocrine disruption? It's pretty well known now. So a few, actually. Okay. So endocrine disruption occurs in living organisms when pollutants, anthropogenic pollutants or natural hormones interact with your body's biochemistry. So where do these pollutants come from? Well, it was first noticed in wastewater effluent, where wastewater has natural hormones that people excrete. There are also soap byproducts, the Teflon from your cooking pans. What else is there? Number of different chemicals that end up in wastewater, and they're not broken down entirely during wastewater treatment, so they get back out into the water cycle. So this has been documented for decades, but it really wasn't a mainstream topic in the United States until the 2000s. This is a very interesting study that came out showing the effect of endocrine disruption on a fish population, where this compound E2, which is the synthetic estrogen found in birth control, was dosed into a lake at environmentally relevant concentration, so nanogram per liter. And what was observed was that the fish population started to die off. Okay, they weren't being born as this chemical was seeded. And then the population of the older fish changed. So in my graduate work, what I did under Dr. Bob Arnold was we went out and we sampled wastewater treatment facility effluents and effluent impacted water as well as groundwater, and we measured these estrogens. And all that work is published. You can ask Dr. Arnold what it means. But since then, I have continued to look at specific chemicals that can be endocrine disruptors. So in this paper published last year, I looked at, me and my student, we looked at a series of bisphenol A replacements. BPA is a chemical of concern. It interacts with basically a lot of nuclear hormone pathways in your body. So when we looked at all of the replacements, we saw that these chemicals act as estrogen mimics. That means if you're exposed to these chemicals, they can induce like a female response in living organisms. And that response could be these fish turning intersex or these fish producing eggs. So this is a bioassay I use, and we're able to compare these bisphenol chemicals to a positive estrogen. Okay. But what we also noticed was that these chemicals are also anti-androgens. So what does that mean? So a man is dominated by the androgen system in sexual differentiation, and women are more responsive to the estrogen pathways. So we also have a bioassay to measure androgenic chemicals. So what we found was that these chemicals will suppress the signaling pathway when it comes to expression of, in response to testosterone. So things that would lead up to male differentiation. Okay, so these chemicals kind of act as a double whammy. Okay, they're estrogens, so they'll induce female characteristics, and then they'll halt male characteristics. In my research, we're also looking at in silico, so computerized modeling of these estrogens with chemicals. So we just construct these chemicals on the computer, pop it into one of these programs, and we can see how these chemicals interact with these nuclear receptors. So it's not enough to show that, okay, we have chemicals that will halt a process. We can show biochemically that these specific bisphenols interact with the ligand binding domain in places where testosterone interact as well as the anti-androgen interact. And estrogen and androgen systems, they've been studied for quite some time. So a new area that I'm getting into is metabolic disruption. So in this area, we're looking at environmental impacts or environmental chemicals that would cause diabetes, hypertension, obesity. And so some known metabolic disruptors are arsenic and bisphenol A. These have been shown to induce diabetes-related ailments. And so what we're doing up at ASU is I partnered with a zebrafish biologist, and we're looking to see how different pollutants affect zebrafish. This is a good model organism. Their homology to human nuclear recepting, this is called a PPAR pathway. Their homology is pretty high compared to humans, so it's a good model organism to study. So what we're doing is we're exposing zebrafish to different chemicals. We have a positive chemical, this is rosiglitazone, as well as bisphenol A. We're looking for production of fats within the fish. So what we have shown is that bisphenol A does induce lipid production in these zebrafish compared to the positive control. So why is this important? Well, we're exposing these fish as soon as they're born. Okay, so these fish are exposed to a known pollutant, and right off the bat, they're already expressing more of these chemicals. So we want to look at additional pollutants as well as polluted wastewater to see if there is a correlation between pollutants and metabolic disruption. Okay, my next area of research is antibiotic resistance. I started this research here as a postdoc at the University of Arizona. And you can see the statement, the wide application of antibiotics in human and veterinary medicine has led to large-scale dissemination of bacteria resistant to antibiotics in the environment. This here is a petri dish of E. coli grown on chloramphenicol. And in this experiment, I was able to show that if you insert a gene into E. coli, we can make it resistant to an antibiotic. So I worked in the lab of Dr. Megan McAvoy in biochemistry and Chris Rinsing in SWESS. And they are well known for their work in metal resistance in E. coli. So between the two labs, I was tasked with a couple of objectives. One, we're looking at the process here of how E. coli are resistant to copper and silver. So this is the outer membrane of E. coli. This is the inner membrane of E. coli. And E. coli has the machinery to export copper and silver from the cell, rendering it resistant to these toxic ions. So this is a large complex protein spanning inner and outer membrane. It has an inner membrane protein, an outer membrane porin, and then a membrane fusion protein. So I was responsible for looking at this little protein, and I was looking for interactions between the two to kind of uncover the pathway of this mechanism. I was also looking at other metal-resistant systems and gram-negative bacteria, and really looking to see if these metal-resistant mechanisms are also responsible for general antibiotic resistance, and we were able to show that. So the protein structure I showed in the last slide is homologous to this one here. Okay, this is the major multi-drug efflux protein in your gram-negative strains of bacteria. And this is what makes E. coli, Salmonella, Pseudomonas resist many different antibiotics. This is an antibiotic efflux pump. It's called the resistance nodulation division. What happens is as soon as bacteria experiences or is exposed to any one of these substrates, it will pump it from the cell. Okay? And this requires some energy. There are different pumps that require ATP or translocation of ions. This one here is a proton drug antiporter. So a proton will be taken up as a drug is expelled from the cell. Okay, so like I said, this is why E. coli and Pseudomonas are so resistant to many different types of drugs, the major multi-drug resistant protein. You can see that it, this renders E. coli, it allows E. coli to survive your gut. It will just export bile salts, many different detergents, metals. Pseudomonas is a bit more robust. I would say it has probably now close to 20 of these large proteins that simply export the bacteria. So in my transition from postdoc to faculty, I came across some literature that showed that these pumps are also resistant to hormones and study hormones. So I thought, are these pumps, are these bacteria resistant to these problematic estrogens that survive wastewater treatment? And so that led me to a couple NSF awards to study the mechanism of attenuation during wastewater treatment. And in this process, we're proposing that your E. coli or other bacteria that are found in the wastewater bioreactor are simply exporting a lot of these chemicals. So they don't have time to oxidize with the enzymes. They don't have time to break down these chemicals during wastewater treatment. So we're looking at the efflux of your hormone from the cell, and then it just ends up back in your wastewater effluent. Okay, so to test these, this hypothesis, we looked at the induction, gene induction of these R&D type transporters. So we looked at 2 strains of bacteria, E. coli and Pseudomonas, and we exposed them to the synthetic birth control pill, the detergent byproduct, and bisphenol A. We wanted to see if any of these antibiotic resistance genes were induced just due to exposure. Okay, and we found that. So what are these genes? So we have 6 efflux genes in E. coli, and the major efflux gene is ACR-AD12C. This is an intermembrane protein which was overexpressed when exposed to these estrogens, as well as this gene. This is an anaerobic efflux pump. Okay, same with Pseudomonas. Okay, basically most of the our efflux pumps were exposed, were expressed when exposed to these estrogens. Okay, so what does that mean? Well, these wastewater bacteria, they're exposed to not only hormones, they're exposed to antibiotics and other chemicals. Okay, but as soon as they see these, they're going to start making these antibiotic resistant proteins. And then they'll just start to export any organic they don't need. Okay, so this was the first step in our test. And so then that led to, so that was just a really short-term grant. And then I got another NSF grant to really look at the process. So this is ongoing. This was awarded a couple years ago. So we're going to look at which wastewater pollutants are substrates of these bacteria transporters. And we're going to do that a couple ways. We're going to look at pure strains, mutated bacteria, and then we're going to add something called an efflux pump inhibitor, where we stop that process, and then we'll look at the mass spec fingerprint with and without that efflux pump inhibitor. We're also going to look at what else we looked at, the prevalence of these efflux pumps in wastewater bacteria. So I had proposed to do qPCR, but since technology has changed rapidly within the past few years, we're switching to metagenomics. And I'll show some data in a little bit. And then finally, we'll look at the degradation of environmental estrogens during wastewater treatment. So I partnered with a chemical engineer at ASU who studies metabolomics. And then we're also applying efflux pump inhibitors to see if we can further degrade, if we stop that process, if we can further degrade these hormones in bacteria. Okay, switching gears a little bit. This is a USDA-funded project where we're looking at antibiotic resistance genes found in final products coming from wastewater treatment facilities. So we're looking at tertiary treated water as well as biosolids. So we want to see what is the prevalence of the many different types of antibiotic resistance genes during wastewater treatment. And what are the implications for groundwater recharge as well as for reusing these products, either for irrigation or land application of biosolids. So this is a small treatment plant represented by this data here. It's standard, primary, secondary. The tertiary treatment is a filter followed by UV and then disinfection with chlorination. Basically what we're seeing is that in the tertiary treated water, we cannot culture any of the bacteria, but we're still finding a lot of their genes in water. With your biosolids, we're still finding a significant amount of antibiotic resistance genes in that product. So it'll be important to see what happens after this. I believe this treatment plant gives their biosolids to one of the farms in West Phoenix, and it's applied uncomposted, okay, so as wet biosolids. I don't know what they use it for. Okay, and then going back a little bit to proteins, I do a lot of work with proteins and protein isolation, protein production, manipulation of the proteins. This is a work by my most recent PhD student where we looked at one of these E. coli transporters. This is the cut protein, which transports potassium. It takes up potassium and is involved in ion balance. But what we also found was that this protein has a higher affinity for cesium. And cesium, radioactive cesium, is a problem worldwide. And so, oh no, okay.
My apologies for the interruption, folks.
Great. So my grad student, she made basically like a very basic artificial cell. Okay, so this is a lipid membrane, and she purified this protein and inserted it into this liposome. And we did some experiments with cesium. She imaged this under cryo-EM, and these liposomes are around 100 nanometers. And we're able to show removal of cesium from water. So this is a novel technology, and why is this important is we can remove cesium at ppb levels. Okay, most technologies require ppm level removal, including ion exchange. And this is because our little protein here, oops, sorry. Okay, our little protein here is a proton cesium antiporter. So all we have is a proton gradient or a pH gradient that facilitates uptake of cesium. Okay, and that manuscript is under review. Okay, finally, this is some newer work that's not really published yet. This is wastewater-based epidemiology. I consider this very, very interesting, very important. Wastewater-based epidemiology collects sewage from a known community, and we can measure different metabolites that have passed through the body or compounds that are just released by the body, and that'll give us an indication of population health. Okay, so this is the process here. If you ingest something, excrete it in your waste, we collect that. And then you prep your sample, and it's best, these compounds are best measured under LC with TAB and mass spec. We're using the presumption that urine is 1% by volume of wastewater. And also knowing the number of people contributing to the sewer line and sewer flow, we can get sewer fluxes. Okay, so we are applying this study to tribal communities where there is a substance abuse problem. So this is a dynamic issue in tribal communities. It affects families and culture. We hear about the opioid crisis nationwide. And different studies have shown that tribal communities are disproportionately affected by substance abuse compared to the national average. Okay, so why look at sewer-based epidemiology as opposed to traditional surveillance? Well, we really don't get a picture of specific tribes. Okay, tribes are different even within a specific reservation boundary. There could be different issues going on. Okay, but we do have access to some databases. We're working with tribal epidemiology centers in the Great Plains as well as Intertribal Council of Arizona. So they do have access to look at substance abuse, either from death certificates, overdose deaths, or from IHS facilities, where they can look at prescription levels, or they can look at drug reports. Okay, so surveys and databases, they don't really show the entire picture. We don't have that high resolution. We have low sampling rates, and it's really generalized for all of US. So with our method, well, so this has been done before, sorry. It's been done, wastewater-based epidemiology, I would say really got its footing in Europe and Australia, where drug use in major cities is tracked. There have been some studies in the US, and I did a preliminary study in Salt Lake County where we looked at the 7 major sewer trunk lines coming into the Central Valley Water Reclamation Facility. We were able to measure oxycodone, THC, codeine, cocaine, and methamphetamine. And we didn't really see strong correlations with the population, but we did see more marijuana use in the rural areas. And then we also saw greater prescription drug use in more affluent communities. And also in Tempe, my colleague, Dr. Halden, is looking at, he's partnered with the city of Tempe to look at substance abuse in that city. Okay, so why should we use wastewater-based epidemiology? Well, there's the anonymity. We don't have invasive biospecimen testing, which is important in tribal communities. We can't really just go in and say, give us a sample. That's often tribally prohibited. This is near real time, so we can go in and get a 24-hour snapshot of substance abuse just by setting up an auto sampler. Once we have data, we can target education and diversion programs to certain areas. And this will complement existing surveillance. So remember that surveys have a low response rate. Here we can go into a community and actually see what people are doing. Some disadvantages, community level use would be generalized. Okay, so if I go into a tribe, we tell them we have net use. Some people might take offense to that stigmatization. Monitoring can be cost prohibitive, because we're using mass spec LCMS. We often need a technician, and then the instrument itself is very expensive. We need a good population count for data interpretation, which is also a limiting factor in tribal communities. Aside from the census, it's often hard to predict the actual number of people living in a community. Okay, so taking this further, if we do take this into, if we want to use this in tribal communities, is that applicable? So we have to look at the wastewater treatment facility. Many of these places are remote and don't have centralized sewer systems. We need an actual sewer system to collect the sample. And then it also has to be accessible. Okay, finally, is the sewer collection representative of the community? I'll give an example. If we have a tribe with a casino, you have a lot of people coming into tribal boundaries that are not native to that tribe and could contribute either positively or negatively to that drug signature. So we shared this work with the head of Indian Health Services, and he said, why don't we just do this for all tribes? Okay, we want to get a grip on the substance abuse problem in our tribal communities. And we said we can't really do that because we don't know what's going on with sewer systems. But EPA does give out sewer permits, NVIS permits. So we went to the EPA website, and we're able to find tribal communities with NPDES permits, and that will tell us where we can start to maybe implement studies for substance abuse. Okay, and those are all highlighted here. Okay, but further, we still have to work with the tribe as well as Indian Health Service. So this project we started in 2015 and going through all the different approvals, finding funding. We're just now collecting data 4 years later. So you need tribal IRB or tribal council approval simply just to conduct this type of research. You have to have your approval and tribal requests laid out before you start sampling. Do they own the data? Do they own the sample? Is this to remain anonymous? Do they not want this published? All of those have to be worked out before we start sampling. Since we're dealing with sewer systems in tribal facilities, that's often operated by Indian Health Service, unless the tribe itself has designed their own infrastructure. Okay. Then finally, your own institutional IRB, you have to go through those approvals at ESU. Okay, so we did conduct a study of several tribal communities. So we scheduled with the tribes, and I won't name the different tribes or tribal communities because they have chosen to remain anonymous, but we work with their public utilities or Indian Health Service. And we take precautions when working with raw sewage because we're dealing with human pathogens. So we need immunizations. We don't allow tribal personnel to handle the sample. Personal protective equipment as well as training in confined spaces. Depending on where we sample, whether it's in Arizona or if it's outside of Arizona, the sample has to be shipped on dry ice. If it's local, we just drive it into campus and start processing it. And then organics are extracted from the wastewater. Okay, so this is a sample setup. We're using a composite sampler, and we're collecting from sewer lines, so it's suspended in the sewer system. And it has, we measure the tube, and then we program the auto sampler to collect 100 mils every 15 minutes for 24 hours. Okay, so once we have processed the sample, quantified on LC at tandem mass spec, and then we factor in any concentration factors, and then we look at the sewer flow rate as well as the population, and that can give us a per capita drug use amount. Okay, so what are the metabolites that can be measured? A number of different metabolites. On the left gives the general chemical class. So over-the-counter, prescription, illicit, cultural, and then all of the different chemicals such as methadone, as well as its metabolite. So what's nice about this process is that we can differentiate between what gets dumped down the sewer and then what has been ingested by a human, because when a human takes a drug, it gets converted into its metabolite. So the metabolites are listed in parentheses. Okay, some compounds of interest. We're looking at a lot of opioids. We're looking at opioid withdrawal medications, alcohol, nicotine, and caffeine, and then some more of the illicit drugs. Okay, so looking at some results. So these plots show the concentration in the original sample of the substance on a log scale. Okay, so some of these vary significantly from community to community. What was interesting is that, well, what was not interesting was that we did find drugs in basically all of the tribal communities. But what was interesting was that one tribe requested monitoring of dextromethorphan, and that's found in cough syrup, okay, over the counter. The tribal drug tester knows that people come in, and when they do their drug testing, they're not, whatever they're taking is not showing up in their urine tests. And so he suspected it was this, and we found dextromethorphan, okay, hundreds of nanograms per liter, which I consider a very high concentration. ADHD medication was confirmed in communities with large student populations, so large high schools. Meth and cocaine was found. We did not detect PCP, LSD, or ecstasy. For the opioids, a community member wanted us to look for tramadol. And it was interesting because the IHS director said that one of the IHS facilities had one of their staff had been shoveling tramadol on the side and was known to be distributing it in the community. And the community members knew this. Tramadol wasn't really ever on our initial analyte list, but I think we found it microgram per liter range in this work. Fentanyl was found at one tribal headquarters. This one is concerning, the opioid withdrawal medication, methadone, which is given to people who go in for drug treatment. Okay, that was detected, but not at levels comparable to, I guess, urban areas or people with the health infrastructure. So what we're thinking is that, where there is a high opioid abuse epidemic, these individuals are really not getting the treatment that they should be. We saw very, very high morphine, but that tended to be in communities where there were IHS facilities where they actually would use that type of drug in their healthcare system. Okay, illicit drugs, so alcohol is found in all communities, and then nicotine and caffeine as well. So this is nicotine, the metabolites, caffeine, and its metabolites. What's interesting about looking at these types of drugs is that these can be used as population biomarkers. Okay, so if we don't have a population count, we know that adults consume X cups of coffee per day. By the time it's metabolized and excreted, we know that what that concentration is. Okay, so in this way, we can measure the population without going door to door and getting that number there. So this study is ongoing. We're currently looking for funding. We're going to NIH. And then we're also recruiting tribes, and we have tribes at different stages of this process. For one tribe, we're still going through that initial agreement. For another tribe, we're going to look at their sewer infrastructure, and then we're collecting samples in another tribe. Okay, and so thank you for listening. So I'm going to continue working with wastewater-based epidemiology. Next up is metabolic disruption of reclaimed water, and then antibiotic resistant genes in reclaimed water, in addition to my NSF-funded project of looking at the bioreactor processes. Many people to acknowledge: my family, my lab, and then my collaborators' lab, the tribal epidemiology centers, the Maricopa farm, as well as the community college, and then my funding. And I'll take any questions if you have.
I was interested in one of the slides that you presented at the beginning,
Okay.
in which you performed some in silico binding affinity test.
Yeah.
Can you tell briefly a little bit more about this?
So this program is called Virtual Tox Lab. It's a series of 12 nuclear receptors. So estrogen, 2 estrogens, androgen, the PPAR system, which is the metabolic system. There's some liver enzymes. And this is the platform in Switzerland. You get a free academic license, and you really only need to know how to draw it on a computer. And then you input the chemical, and it'll come back about 24 hours later. You'll get a binding affinity for any of the proteins you want to see if it binds to.
Very interesting. Thank you.
Yeah, it's a first screening process, first step in the screening process. And you can look at even unknown chemicals if they interact with nuclear receptors. A question, Bob?
Yeah, in light of the sizable opioid settlements that are on the horizon, is there any communication from drug companies with either you or any of the colleagues that you know are working with opioids, so they can sort of gauge their viability down the road?
Not that I'm aware of. I mean, they haven't contacted us.
Second, there must be a lot of parallel investigations with non-tribal communities.
Yeah.
So have you had the opportunity to compare the outcomes?
So we're still looking at sewer flow data for these results, but the concentrations found in tribal communities are higher than in urban areas, just based off literature review. But these tribal communities do have lower sewer flow rates. They don't have that large dilution factor that an urban area might. So we really need the population and sewer flow data.
Are social services agencies able to use this information to target some community-based intervention or education programs for the drugs?
Yeah, so that's what we're hoping. With one tribe, we have reached out to the substance abuse director for that tribe, and he's very interested in this project. And then we're still working with the other tribes to see what level of interest is this. Some communities want their tribal members to see the data, to see what is in their sewer, and that we can see what drugs are using.
Because a lot of these are prescription drugs. And is it easier or more difficult in a tribal community to control the volume of prescription drugs which leave the doctor's office? I guess the question is it more centralized in terms of how people get their medications, or is it more generalized?
Yeah, that I don't know. Our tribal epidemiology partners are going to look into prescriptions and then compare that to, I guess, nationwide usage. But I mean, just what I know from certain tribes I work with, it's pretty easy to get prescriptions and then abuse.
Following on this, couldn't this be the issue with methadone and Suboxone is that not a lot of doctors are allowed to prescribe it, and the cost this medicine is around $600 for a month's supply.
Right.
Yeah,
IHS
unless you get a
facility.
coupon, which is still $150 with a coupon. Like, is there any way to have more doctors who are allowed to, because any doctor is allowed to prescribe opiates, but they're not allowed to prescribe methadone. Kind of complicated. Maybe try and bring this into hospital centers,
Right.
reservations.
Well, it was found, so I mean, it's used
Yeah.
right somewhere. But I don't know how it works with it, as far as their prescriptions and then the cost of it.
Concerning your antibiotic resistant pumps in your E. coli, you said that you have an antagonist for it. How does that antagonist work?
So people are still researching that, but they're basically large organics that can either block the channel, so the pump goes through like basically a funnel, that can block that, or it can stop that proton translocation. So there are different ways it can happen.
Have you looked at methods for downregulation of these pumps, like antisensorinate?
Yeah, I'm not sure how I would do that.
Are there any online viewers who would like to ask a question? Would please unmute yourself or type it into the chat. All right, that answers that. Questions or comments? Well, I would like to thank Dr. Conroy-Ben for joining us today and presenting.